Hello, my name is Shriya Thakur and I am going to present positive and negative monochrome staining. We started with introduction to staining. Staining is basically coloring microbes with dye to observe under microscope. It enhances contrast between organism and background. It also helps to observe or identify morphology, size and shape and arrangement of microbes. What is monochrome staining? Monochrome staining is a technique in which only one dye is used to stain microbes, mainly to study their shape, size, and background. It does not differentiate between types of microbes but increase contrast for easier observation. It means monochrome staining is staining technique which use only one dye and it mainly used to study their shape, size, and arrangement, but it does not differentiate types of microbes, but it increase contrast between microbes and background. Principle is based on the interaction between the dye and the cell surface, which is two types of interactions: basic dyes and acidic dyes. Basic dyes bind to the negatively charged bacterial cell wall. Cells get stained. Acidic dyes do not penetrate the cell. Background gets stained. Cell remain clear. It means in basic dyes only cell gets stained but background remains colorless, or you can say clear. In acidic dyes only background get colored but cell get colorless. Definition of monochrome staining. Monochrome staining is the process of applying a single dye to a microbial smear in order to increase contrast between the cells and the background so that the shape, size, and arrangement of microorganisms can be observed under a microscope. Now, positive monochrome staining. Definition of positive monochrome staining is that it is a type of monochrome staining in which basic dyes bind to the negatively charged component of microbial cells, making the cell appear colored while the background remains clear. In this, only cells get stained and backgrounds remain clear or colorless. Procedure of positive monochrome staining. Take a clean glass slide, add a drop of bacterial culture into it, then aerate the smear, heat fix the smear, and then fold the slide with a basic dye, for example crystal violet, safranin, methylene blue, for 30 to 60 seconds. Then get gently washed with distilled water, blow dry it, and observe it under microscope. Advantages of positive monochrome staining. It is simple and quick. It requires only one dye and minimal steps. Inexpensive. Basic dyes are cheap and easily available. Highlights morphology. Shows the shape, size, and arrangement of bacterial cells clearly. Good contrast between colorless background, or you can say clear background, and microorganism. Easy to perform for beginners. Often the first staining method taught in microbiology lab. Primarily, identification provides basic information before complex staining. Permanent preparation. Since the smear is heat fixed, the slide can be preserved for longer observation. Now, negative monochrome staining. Definition of negative monochrome staining. Negative monochrome staining is a staining technique in which an acidic dye is used to stain the background of the slide while the microbial cells remain unstained and appear colorless. Common dyes, example nigrosin and India ink. In this, only background gets stained but microorganisms get colorless. microorganisms remain colorless. Purpose of this is that it for to observe the true size, shape, and arrangement of microorganisms, especially delicate structures like capsules. We observe specially structures like capsules from this staining method. Now, procedure of negative monochrome staining. Take a clean glass slide, add one drop of bacterial culture into it with dye, and use another slide to spread the mixture. Place the edge of the second slide at an angle, touch the drop, and pull it across the slide to create a thin film. Then air dry the smear. Do not heat fix. It is very important to do not heat fix in negative monochrome staining because heat can distort delicate structures of cells like capsules, etc. Observe it under microscope. Advantages of negative monochrome staining. True cell morphology. Cells are not heat fixed, so their shape size remains same or undistorted. Capsule visualization. Special structure. Delicate structures are visualized. Note cell damage. Since the dye does not penetrate the cells, fragile microbes remain intact. Or you can say sensitive microbes remain intact. High contrast. The dark background makes the colorless cells stand out clearly under a microscope. Simple and quick. It requires minimal reagents and easy to perform. That's why it is a primarily technique. Useful for small or transparent microorganisms or organisms that are otherwise difficult to see with positive staining become visible. Here is a animation form of procedure of monochrome staining. First, spread a culture in thin film over slide, then air dry it, heat fixing, and then fold the smear with basic dye and gentle wash with distilled water, and lastly air dry it and examine it under microscope. Conclusion. Monochrome staining uses a single dye to visualize microorganisms and their morphology. Positive staining and negative staining. Positive staining cells take up the dye, appear colored against a clear background, useful for observing shells, size, shape, and arrangement. Negative staining dyes stains the background, leaving cells colorless, preserves true morphology, especially delicate structures like capsules. Both techniques are simple, quick, and essential for basic microbial study, providing foundational knowledge before more complex differential staining methods. So that's all about monochrome staining. Thank you.